Great, great, great. Thank you. Thank you so much. Just a little clarification on that title. I meant metabolomics in a broad sense. So not just metabolites or collections of metabolites, but metabolism and everything related. So I'm delighted to be here. dkNET is exactly the sort of resource suite that I think every community needs, not just one, but probably 3 of these. They're a challenge to put together, I gotta say. Many of you I know have been doing this for years, Marianne and team at NIF, Jeff, Nita. These things are beasts to maintain, feed, and they're never done. Doing web service development is like, in some ways, like being a shark. You can never stop. There's never a moment for relaxation. So this is the abstract that if you bother to read abstracts for talks, there it is. I will come back to those top 3 questions here toward the end. I do have enough time that I think if you see any burning questions in the chat box, please go ahead and just interrupt me and I can probably answer them, especially if they're questions that really would benefit from immediate clarity, like which button did you push exactly, or could you go back 1 slide? I'm happy to do that. So please do interrupt if necessary. Yeah, so the talk is going to be divided into 2 or 3 main parts. So I'll call it 3. The first is going to be a rant by me on fair data, good data, junk data, and what we as a community of scientists should have been doing, frankly, for the last 50 years, but haven't been doing because of our good, but not all obsessions are inevitably good, but we've been obsessing for the last 50 years on reductionist paradigms. And that is a bad thing if you're trying to translate to very complex human populations, because humans are anything but reduced. They have an enormous amount of developmental complexity, genetic complexity, exposure complexity, social complexity. And unfortunately, each one of us is an N of 1 experience, unless you happen to have an identical twin. I'm going to be talking a lot about getting beyond N of 1 science, using mice and rats, but everything I say also would apply to humans with some caveats. I do want to point out at the get-go that mice and rats are, believe it or not, supraprimates. Yes, mice and rats are supraprimates. They are a member of the Euarchontoglires. I don't even know how to say that, but the normal name for that is the supraprimate superorder. And our other supraprimate members are lagomorphs, Coluga, tree shrew, and as you can see, the not so great apes are also included. So if you happen to be using rodent models in your work, do not feel like you're not doing the right thing. You are definitely doing the right thing. They're really quite relevant in an evolutionary context, and they're great for experimental control. And the huge advantage of mouse and rat is that you can inbreed them. That also can be a little bit of a risk because some people only study one inbred strain of rats, and that's what I'm going to be ranting about here in the first third. And I just want to thank my colleague Yvonne Gerling at the University of Tennessee Health Science Center, who's been my mentor in anything having to do with diabetes. I'm definitely not a real card-carrying metabolism researcher, although I've been doing it for a while. And I'd like to also thank NPOD, which has also provided context for some of the work Yvonne and I are doing. So I'll spend a minute on this slide. Sorry for all the words, but it's important for all of us. There is this tension I already mentioned between reductionist approaches and more integrative, holistic approaches. I can remember how horrified I was in about 2004 when Elias Zerhouni, the then director of NIH, said, "Thou shalt translate." And all of us doing basic research were aggravated because we thought we knew how best to address human health and disease. And being told by the director that we should translate our findings seemed like an imposition and a vast assumption that that would really help. Turns out he was absolutely right. Translation is vital if you're going to get money from the taxpayers. And we as a community, again, do a lousy job at it because we typically use just one strain of mouse or rat. Synthesis is critical. So I think in 2004, you could argue that computer resources weren't quite there yet to do heavy duty data integration. We don't have any excuses anymore. A petabyte, I could have a petabyte of memory at home. It set me back $50,000, but I could do it. The computational resources for doing heavy lifting, you do not need a supercomputer. You can usually do it on your laptop if you do it in a smart way using modern coding techniques. We know this for a fact. Theme 2, human disease is almost entirely due to complex interactions. I've already said that. Do we have a solution? No, we're like total babies. We're pathetic. GWAS is in some ways pathetic. GWAS does nothing with the environment. It just throws the environment under the truck, under the train, under the bus, whatever you want to throw it under. GWAS throws environment under the bus. It ignores it. It provides population level estimates of genetic effects. That's all it does. That's not going to do you any good if you go to your physician and say, "Hey, I think I'm pre-diabetic. What should I do about me? How should I be treated?" Because if your doctor then says, "Oh, here's your polygenic risk score given the 500 loci we have for diabetes, you're at high risk or low risk," that doesn't mean anything to you. You knew that before you walked in, or you could have known that before you walked into the doctor's office. So when you read about polygenic risk scores and how excited the GWAS community gets about this, please, big grain of salt. There is no big E in the GWAS equation. So is there a solution for that? Of course there is. And I'm going to tell you what that is. Theme 3, okay, so here is the solution. We need to acquire the right type of data very systematically, not hit and run data, not I'm going to get my R01 next week. I'm going to spend 5 years getting data, then I'm going to throw the data away unless NIH insists that I store it somewhere for 5 years, and then I'm going to start all over again. Rinse and repeat for the rest of your career. Throw all of your data away. That is, I would use a word that starts with the letter R, but I've been taught by my medical students that I should not use words that start with the letter R. So I'll just say it's a very neurodiverse approach to data handling, and thank God for FAIR, and thank God for just better ontologies that make it possible and bigger hard drives and SSDs that make it possible to save data and to go deep and to accumulate data. I don't want to stand on any shoulders of giants. I don't believe there are any giants. I want to stand on a huge mountain of data, and I don't care who generated it as long as it was well generated. So that throw away that standing on giants idea. That's, it might have worked for Max Planck or Newton or whoever, but it's a silly idea at this point. Okay, so I'll be covering this solution pretty well over the next few slides, but that's most of my rant. I'll probably come back and rant a little bit more, just because I like to rant. I don't know why. As I get older, I like to rant more. I used to be more mellow, as Marianne could maybe tell you. I don't know. It'd be interesting to know. So having made or just pointed out the tension between reductionist approaches and holistic integrative approaches, obviously we need to bring the two worlds together. Reductionist approaches can be awesome. They really can get us to these things. Unfortunately, we use the word mechanism, completely silly word to use. Does anybody know where the word mechanism comes from? It comes from Descartes in 1680, whatever. Basically, he regarded human bodies as mechanisms, and then you had the pineal and the soul was a separate entity, and the two came together in this magic Cartesian dualism. Sorry, humans are not mechanisms. What happens in a cell is not a mechanism, but we use that word because our study sections like it. What we call mechanisms are actually hideously complicated processes, and usually one mechanism is about 100 processes. So just another little bit of rant. We have to bring these approaches together. Yes, go ahead in your next grant application, use the word mechanism, but know that if I read your grant application, I will roll my eyes into the back of my head and moan silently to myself. These are papers, one that says, let's mutate every gene in the mouse genome one at a time on the background of C57BLAC6. That's kind of the ultimate reductionist paradigm. It's not genetics, it's functional genomics. It's actually worked pretty well. So I would not say it was a bad expenditure of money. It's really had some great deliverables. The first author on this paper in 2008 is a colleague of mine, Johan Oryx. Everybody else is sort of the who's who of mouse genetics in Europe. And then this is Johan after 11 years of exposure to me, writing a paper with my son on bringing more holistic approaches together with reductionist approaches. So kind of a nice pair of papers for those of you who want to see how you can bridge these methods. I did want to give you an example because many of you are diabetes, metabolism experts. You will have heard about polygenic risk scores and how cool they are and how they're going to help. I think read Mike Snyder carefully. He has had some brilliant papers on what precision medicine is going to mean for pre-diabetics, since he is pre-diabetic himself. Mike is doing it the right way, time series data for individuals, and then estimating the risk score for that individual based upon that individual's data. And yeah, the polygenic risk score might get in there a little bit, but it's not the only thing. So let me show you why I have a gripe with polygenic risk score. So this could be diabetes, but this happens to be for coronary artery disease. A bunch of genes known to modulate risk for coronary artery disease. We take those SNPs, we evaluate the cumulative multiplicative risk. So if you have one gene variant that confers a risk of 2 for developing coronary artery disease over a population of 1 million subjects in England, say UK Biobank, and you have another risk variant that confers a population risk of 0.5, you multiply 0.5 by 2.0, and now your cumulative polygenic risk score is 0 or 1 actually. So you're at the population average. The problem is you didn't think about your environment. And if we now on this second score add the environmental risk score, what's your BMI? Do you exercise routinely? Do you do aerobics? Do you run? Do you live in a stressful environment, or do you live on a golf course? Do you take 2-month vacations or not? Do you ride a horse or not? So all of those subtle factors play into your environmental risk. And if you take this individual A, take the polygenic risk score, and then convolve it with the environmental risk score, you've got a different beast. So that's why it's so silly to think that polygenic risk scores are going to be applicable for individual A or for you as an individual. So now we're getting close to the solution. What about animal models for common disease? And I've already poked fun at this, but it's sadly true. And despite the fact that Francis Collins, who has done a fabulous job running NHGRI and NIH for the last 19 years, he's a really good geneticist, but he's a geneticist who studies Mendelian disorders rather than complex diseases. Now, everything is a complex disease. Even cystic fibrosis is a complex disease. Even progeria is a complex disease. But if you study simple Mendelian diseases, it does put you in a certain mindset to get at gene mechanisms, to get at functional genomics. That's not my mindset. My mindset is to understand architecture of variable traits, whether it's metabolism, insulin expression, glucose utilization, mitochondrial function, et cetera. All of that is complex. And so if you look at the All of Us program, I don't know how many of you have been genotyped, but if you sign up to All of Us, allofus1word.gov, or maybe it's NIH, you can get yourself sequenced for free 30x whole genome. Here's their cross-section of humanity, which makes me laugh because I'm this guy over here now. It makes me sad. You know, the old white guys are supposed to be, they're supposed to be like 10 times as many of those, I thought, but apparently not. So anyway, they did get this photograph right, obviously. There are 9 billion of us on planet Earth. We're each more or less genetically unique, and God help us to figure out what the best course of action for any one of us is in terms of staying healthy for a long time. And God help us doubly because, again, we're studying C57 black 6 over here, or we're studying one strain of rat. I don't actually know what strain of rat that is. It's obviously, it looks like a healthy wild type rat that you might find in your gutter, but most rat work is Sprague-Dowley. So this 95% of more of rodent research that NIH spends on N of 1 experiments, I don't care if your study has 10,000 animals in it. The Allen Brain Atlas has probably burned through probably on the order of 20,000 58-day-old male C57 black 6 animals, and they think they're telling us something about how the brain works. Does that puzzle anybody other than me? I mean, I love the work. I will defend the Allen Brain Atlas, but it aggravates me that not a penny has been spent looking at genetic diversity, almost not a penny. There was one study they did because I utzed them aggressively for about 3 years. They looked at the GPCRs in the brain of 8 strains, and oh my God, was there variability. And then they buried that paper and never thought about it again. Same thing for diabetes, same thing for metabolism. It's 95% N of 1 science. I don't care how good it is, it's not good. It's actually inexcusable. So here is a study, and I'm going to use it as kind of an example of how I think we need to bridge reductionist and non-reductionist methods. I need to share this paper with you. So I'm going to put it in the chat. It just came out in let's see, what do I want to do? I want to stop that, go here, and I want to give you this paper. So here it is. Oops, wrong place for that. So that should be coming at you. It's a 4.3 megabyte paper in Nature Metabolism. This figure actually comes from a work that's in press in Cell Systems that I will also put in. I think it's a little bit bigger. It may be too big. I don't know how this will work in, so it's 40, this is a 40 megabyte proof. So it'll take a little longer to get to you. They're companion papers, same cases in both studies. The first one that I gave you there is easier reading. The second one is more omics. They're both all about metabolism. They don't really have a spin on diabetes, but I think anybody who's interested in diabetes is by necessity, of course, interested in insulin and by necessity interested in general metabolism. So let me then switch over here back to full screen mode and tell you what we did here. So we've generated a family of 150 strains of mice that are a cross between C57 Black 6 on the one hand, the canonical mouse that sucks up about 95% of all NIH mouse dollars, and DBA2J, which is the oldest inbred strain of mouse, actually the first inbred mammal. Inbreeding started in 1911 up in Massachusetts at the Busey Institute. And we basically put those two strains of mice, which are both inbred, into a cage and let them mate, and then took their F1 progeny and then did F1 by F2 by F3 by F20 mating. So filial matings, brother-sister matings for 20 generations to generate a new inbred strain of mouse. And each one of those new inbred strains of mouse or mice is a hybrid between the two parents, obviously, but they're inbred. So each one of these colored mice here, and I wish they came in these colors, they only come in 4 colors, but each one of them is genetically fixed. So there's an orange mouse, a red mouse, a cyan mouse, and they're replicable. So I can make you 500 of these cyan mice at any stage. I could treat them with streptozotocin. I could treat them with high fat. I could do whatever tricky thing you can think of with both genetically identical male and female, just like you could do with Black 6. But now you've got 150 of these you can use. And what we did in the study is we took about 80, it says 89 here, but about 80 different strains, all females, for the reason that it's a lot cheaper to age females because they will live relatively peacefully in a cage, whereas the males will beat up each other. And our vets get very upset about that. And sometimes we do too. And we put 10 of each of these strains on a standard chow diet, and 10 of them we put on the ridiculous blue diet that probably many of you know. And we just determined how long they live and what is their insulin level, what is their glucose tolerance test results. I wish we could go in and fish out the pancreas and tell you what the beta cells look like in those animals. But as you know, pancreas is a major motion picture to do anything with. But if any of you are interested in doing that, I'd be happy to work with you. I know Yvonne would be delighted to work with you. We do have some data on islets that Yvonne has generated, but not enough. So the simple design then is it's a perfectly randomized clinical controlled trial, but using mice where our instrumental variable is the diet you go on. And of course, your genotype is also an instrumental variable. But we basically split half the cohort over here. So roughly 11,000 animals, roughly 1,100 animals, 1,100 animals. Most of those were then just followed through to natural death. And 600 plus individuals were then used for the metabolic profiling. I shouldn't say metabolic because it involves proteomic, metabolomic, transcriptomic. All of these strains, by the way, have been fully sequenced. So we have really good data using linked read technology. In the next year or two, we will have telomere to telomere genome assemblies for all of these mice. And here, since Marianne Martone and Jeff are online, this is probably a slide I probably showed at some Human Brain Project meeting, perhaps 15 years ago. I haven't modified it much, but I should, for this audience, obviously, pretend there's a pancreas there. This is a paper that I sent you, the first one that's the shorter of the two, same protocol. And now you should understand the resource. And now it's time to actually show you what it looks like in real life as a fair web service. And I wonder, let me just check what my next slide is. But just have a look at the very bottom of this slide. Data gets better with age. That is rarely true. Most data evaporates with age. These data will get better with age, absolutely. But I would say, again, 99% of data that's collected as part of R01s evaporates. It's so sad. Not only is the design not very good usually, but it also is evaporative data. Evaporative data is, I shouldn't use the R word, is not good data. So what is the solution? And this gets to the theme of so much of the dkNET. So smart data, and I'm going to call it quadratic data, or you could call it just multiplicative data. It's way more than quadratic, though. It's more than power of 2. It's actually exponential data, so smart exponential data. And what it means is, first, what is it? First, it has to have great metadata. Data without metadata is junk. How many of you have gotten Excel spreadsheets from your colleagues that had no metadata at the top, come back to that spreadsheet in 2 years and gone, what was this data again? Is this mouse data? Is this rat data? Is this? So the metadata. So in my advice, if I ever get an Excel spreadsheet from anybody, I ask them, put 10 lines, put 10 rows of metadata at the top. Tell me what your column headers mean. If you want a data dictionary, fine. But if you don't, just put it in there. So good metadata. And all of you on this thread have heard this innumerable times. Metadata is the bane of most biologists, you would think. Of course, the data has to be QA'd and QC'd so that you can wrangle it into a database and clean it up. And this is apple pie and motherhood to many of you: findable, accessible, interoperable, reusable. But that's not nearly enough. It's a good start. Fair data, if you put the metadata in here, if you said it has to have great metadata also, that would be good. But even that's not nearly enough, because if you want it to be really reusable, replicable, I want it to be replicable, not just reusable. I want to know that I can replicate the work that you did 5 years ago. And if you changed your operating system or using a different version of R, or you're using the latest version of Python 3.6, and yet all of the work was done with Python 2.8, I can't replicate your workflow. So it's really important that the entire code stack, and for those of you like Jeff, who are intimately familiar with code stacks, you'll know that that is a very heavy lift. To say I can come back in 10 years to the code that we're running today in a container and verify that everything in that container is the same as it was 10 years ago, that a cosmic ray didn't do something, that's a pretty heavy lift, but we have to get there. We need entirely replicable software workflows that are persistent, ideally for at least 100 years. Then we'll have true fair plus data, where the plus simply means that the entire workflow is good. But I want to just also point out, Marianne and I learned this the hard way in dealing with Google back more than 15 years ago. We were talking, you know, we wanted Google to help us on the big issues in big data. And they didn't want to have anything to do with ontologies. they just, you know, to them, that was too much work. I think they must have read Rob Pike's book on programming. So data dominates. If you've chosen the right data structure and organize things well, the algorithms will almost always be self-evident. Data structures, not algorithms, are central to programming. And that's true of science too. If you generate your data using a lousy process, it'll be lousy data. If you generate your data using a good process, it will be great data, or could be great data. And any N of 1 data set, unless you know exactly why you're doing it, is not going to be great data. And any data that you can't come back to and use as part of a workflow in 10 years is, by my definition, not great data. It's okay data. So that leaves the question of what the heck is the multiplicative part of this doing in this question. And multiplicative means that if I generate a vector of data, my right fingers are a vector of data. It's 100 scalars deep. And in my left hand, I have another vector of data, 100 scalars deep. Can I do anything with these two vectors? And if this vector was acquired in C57 black 6J at 58 days in a male, and this vector of data was acquired in Balb CJ Bailey in females at 300 days, then these two data sets do not talk to each other. There's a hard confound. So you have to, what we want is vectors of data that we can basically use to form an XY coordinate representation, a scatter plot, if you will. So that then becomes multiplicative. And so it's really important to me, and has been in my career, to make sure that almost all of the data I generate are multiplicative. And let me show you what that looks like. Here's data on longevity from the paper I just showed you. These are data for about 80 strains of the BXD family. Some of them live only to be 315 days of age. They die of cancers. Some of them get out to 900 plus days. And now I can take that vector of data. So I'm just showing it to you here as a histogram, but I can take it as a vector, and I can compare it to millions of other traits we've generated for the same BXD strains of mice. That's multiplicative. One vector times a million other vectors. It's not just quadratic, it's exponential. And I call that, so if I just did this for one environment, I would call that a phenome. If I do it for 100 different environments or 2 different environments, high fat and low fat, I call that an oligophenome, 2 phenomes. If I did it for 10 different treatments, it still might be an oligophenome. If I did it for 100 different treatments, now it would be a true polyphenome for this set of individuals. Now I've got the substrate that I need for predictive modeling, for causal modeling, and to provide true precision healthcare, better yet, accurate healthcare to mice. And if I can do this for mice in 10 years' time, then there's some hope that I might even be able to do it for humans, a specific human, like Mike Snyder is trying to do for pre-diabetic people at Stanford, I might be able to pull it off for humans. It's going to require a lot of time series data in humans, which is exactly what Mike is doing, instrumenting everybody up the wazoo for as long as they can stand and for as long as NIDDK will help pay for it. That's the way to do it. But we need to do this in humans too. I mean, in animal models too. So I'm going to now switch over to a live interlude. If I'm looking at the clock right, I have about 5 minutes of live interlude. So please just interrupt me when you think it's a good time to pull the plug and convert over to questions. I'm showing, here's a great paper that I can also put in the chat when we're done. A platform for experimental precision medicine, the extended BXD family. It basically is a kinder, gentler version of me ranting at you about how we should be doing science. I should be ranting at institute and center directors. I do enough of that, but not enough to actually have changed anybody's mind yet. There's a criticism of everything I'm about to tell you is that all of these strains of mice are inbred. Why would we study inbred mice when we can study outbred mice? Humans aren't inbred. Well, actually, we are fairly inbred for a species because at least if we're non-Africans, because of the out of Africa bottlenecks. Africans do not have the same inbreeding. They're about twice as genetically diverse as we are. But it would be good to get off of the not study inbred. So here's, if I made I hope you can see that. Let me see if I can make that a little bit bigger. So here, if we cross DBA2J, now I've lost DBA2J to DBA2J, a male to a female, we end up with a DBA2J. Black 6 to black 6, we end up with a black 6. But we can make the F1s. We can cross a DBA2J female to a black 6 male or whatever. I'm not sure I'm talking correctly anymore. And that produces F1s. The F1s are all isogenic. They're genetically identical, one to another, but they're not inbred. So the idea going forward is not to just play with inbred strains of rats, or mice, no matter how many we have, but to do a massive diallel cross. And with 150 BXD strains here in Memphis, actually about 120 that are easy to breed, that's 120 by 120 diallel cross. That is 14,400 animals that all have perfectly defined genomes. Okay, so now what does it look like in GeneNetwork? And let's answer some of the questions that we had here. So the questions, I'll just put this over here. So what is GeneNetwork good for? So here are the questions that are included, and I'll definitely share this document with all of you. I'll put it in the chat while you're asking questions. But what is the relationship between insulin levels as a function of diet, age, body weight, lifespan in mice or in humans? This is what GeneNetwork looks like, the newest version. And as I mentioned, it did start a long time ago as Portable Dictionary of the Mouse Genome. Was then funded by the Human Genome Project. And we'll go to the mouse data set that I just showed you. I just gave you the paper for this data set. So mouse, National Institute on Aging Longevity Study, traits and cofactors, proteome, methylome. We do have a formal metabolome here. And let's just have a look at the data that I just told you about, which is, I'm going to make this my default. So now you're getting the primer part. And I'm going to do a search for longevity. And I'm going to do a search for insulin for fun. So two terms, if I wanted, this basically treats it as an or, longevity or insulin. This treats it as an and, a Boolean and. I want the or. And I hope it doesn't take too long. While it's thinking, let me show you what should happen. It should get us going to open a backup system. Always have a backup if you're giving a live web demo. There it came anyway. Okay, so that took a little longer because of the Boolean or search, but this is age of death, natural or sacrificed. We don't want the sacrifice date, probably. This is the longevity data I mentioned. Here's what a fair vector of data look like. So I'm going to give this vector to you. I'm going to put it in the chat. That's a fair vector of data. With that URL, which should work, I hope in 100 years, I'm not going to hold my breath. I wish I could hold my breath for that long, but that should work in 100 years. And in 100 years, it should, hopefully the interface will be a little slicker. Maybe general AI will be rummaging around and humans will be playing at the beach. But that's natural lifespan data for 1,700 animals. Here's the distribution of lifespan. It's pretty normal. It's actually beautifully normal. These are the unlucky animals over there. It's exportable to Chart Studio. You can do all kinds of fun things here. It's about as normal as a data set will ever get. So you can do your QC here. You can transform the data. If you said, hey, I want to log to it just for fun, you can do that. Obviously you shouldn't, but you can. So the thing that makes this fair plus is that I can play with the data. I can reset it. I can say, I want to look at only the animals that live over 600 days. I can say, please filter this by sex. I'm going to block all of the females. So now you're looking at just the males. So there's a lot of stuff you can do here. And most fun thing is we can go back and see where's insulin. I can put this in my collection, but we need to find the insulin data. And I realize I'm getting pretty close. So let me just do a search for insulin. I don't see insulin there. We do have some insulin. Let me pick leptin because I see it.
I see it in 54. Isn't that serum
54.
insulin?
Thank you.
Yeah.
Great. I don't know why I didn't find it. Okay, so I've got insulin, leptin, and lifespan, and I'm going to put them into a shopping cart essentially. And I'm going to call it and now I have 3 traits. Oh, I'm a great fan of ontologies. I see Anita's comment. Yeah, they're just, and Anita knows this way more than I do. They're really, really hard. And that's why Google didn't like them, because they thought they were just going to AI their way out of all trouble by just throwing at some wonderful neural network. So here are 3 traits, and I can say, let's have a look at the correlation between lifespan, leptin, and, oh, this is not going to work. Well, it worked better than I thought. So here is lifespan, and here is the correlation of lifespan to insulin. And I'm surprised that worked because the way I structured the search. Let me restructure that and go. I want to get even the animals we killed, so natural or sacrificed. So I'm going to add that to my dkNET collection. Now I have 2 lifespans. I really want to know what's the age at death and its relationship to insulin and leptin. So let's recompute that correlation. There are all kinds of other cool things you can do here. And the correlation we were looking at was age at death and this is going to be insulin. Now this is on 2 diets, but so we're going to come back to this and we're going to say, oh, but we have to take into account the fact that some of these are high fat and some are low fat. But that's a kind of a cool figure you can generate with FAIR plus data. And I can wrap this up into an R-Shiny or a Pluto notebook or which frankly, a better version of a Jupyter Lab notebook, a little more interactive feature set. And you could provide this to somebody in 20 years and they would be able to analyze it. So on this, I forgot to put in treatment. So you say, okay, go back here and find treatment. And Marianne, help me. They're all going to be females here, but I'll just add it anyway. Treatment. Let's just see diet. Okay, so here is the diet variable. We're going to add that into the shopping cart. And I am cognizant of the fact that we're at 1:51 my time and 11:51 most of you California time. So I'm going to quit. I'm going to give myself 1 minute just to show this to you. So we have diet. Oh, no, I'm going to use diet as a cofactor. I'm going to give you insulin and age of death. And I'm going to show you that scatter plot. There it is. And now I want to add a cofactor from this. And I want it to be diet. And now you have the blue and the red where I believe the red is, no, the blue must be the high fat because I think that suppresses insulin. Unfortunately, our code is not quite where we want it to be in terms of 0 equals standard chow, but it doesn't give us the color decoder secret ring right now. Oh, yeah, it does. So blue is high fat and red is chow diet. So there you just do the hover. So this gives you a very baby example of what I mean by smart quadratic data. And because there are so many other factors in here, I could waste my entire Saturday asking and answering questions. These are all the answers. These are all the strains. The same data, by the way, is available at the level of strain means. And let me just close with one more thing to show you some human data. So this is looking at pancreas in human from GTEx. And I'm going to, I guess, leave that to most of you as here is insulin. Let me put this, I'm going to put this, sorry, I'm using the
Yeah, I think we're running out of time, Rob, because we're down to like 6 minutes.
Okay. I'm gonna share.
But I do want to invite you. dkNET has just launched the Hypothesis Center, which provides sort of in-depth tutorials of how somebody would be able to do these steps themselves to answer a set of questions. And so I hope you'll follow up with us so that we can add GeneNetwork to the Hypothesis Center, because I'm of course known about this for many years, and it's an incredibly powerful tool. And this data set in particular just seems like it's got a wealth of information in there. So I hope you'll be willing to work with us to make it accessible via the Hypothesis Center.
Absolutely. And you guys have the software chops and the ontology chops and the RAD chops to make it work.
Yeah, so I think we'll follow up, but I just want to thank you for an extraordinarily entertaining and informative talk. Are there questions from the audience?
I got one question, but I think maybe if there's a tutorial will be easier. So this question from the audience ask, what should they really need to put in the terms in getAny and then in the combine from the landing page?
Yeah, there are some primers. I didn't take you on a tour of the menu bar, but you'll see one of the menu items is help, and there will be some primers in there. Probably not good enough. We're not very good at the outreach because we use this as our own tool. So we tend to focus on making it useful for people that do what I do, rather than people that don't do what I do. So we are going to revise and this is where Marianne and team could really help, because we need to make it more user-friendly, newbie-friendly. But there are some primers, and if you just do a search in the references, there are also some, the top ranked references and the reference pull down will also be mainly primers. And then of course, this video itself might be good, but there are mess of videos that are available from me on using GeneNetwork. I'd be delighted to share with Koei and anybody else who's part of the dkNET.
Thank you. So Rob, I think you started your talk off with translational research and how that was a good thing, right?
Yep.
So intuitively, I've always felt that the approaches that take into account, sorry, my gardeners are coming by right at the wrong time, that takes into account genetic variability and environmental variability would be much more powerful, because that's how we operate in the world. What would you say have been your main translational successes with these approaches, with these?
We've had a couple of lucky ones. I would say our luck rate, though, is probably edging up toward 10%. So the first one, the one that I think is where clinical trials are now ongoing, is in glaucoma. So lucky for us, one of the parents suffers from glaucoma, one of the mouse parents, the DBA2J. And we mapped interocular pressure and got a very strong locus, and we're able to, it's a big calcium accessory protein. It's targeted by gabapentin or pregabalin, and that's a drug that's already in use extensively for epilepsies. So we just made an eye drop, we being everybody except me, made an eye drop version of pregabalin, and it lowers IOP as a function of genotype, as you'd expect, right? You don't expect it to work for everybody. And that is now being slowly, because it's always slow, pushed as a candidate. There are a number of metabolic traits that have been cloned by my colleague Johan Oryx and my son Evan. None of them are yet directly relevant in a clinical setting. I think there's a paper, a diabetes paper that just came out from a group at Mount Sinai. I'm one of the co-authors, the one that knows the least. It's relevant to diabetes or kidney failure associated with diabetes. And that gene may have some relevance clinically in the next 10 years.